Okay. So I'm going into the presentation. So first, I would like to discuss with you about the importance of seed vigour. Okay. So this is small graph which we all used to see when we are taught about seed vigour and viability in our undergraduate course. So you can see this graph. So what this graph implies is the decline in seed viability. Decline in seed viability is later when compared to the vigor. So you see in this graph, this is the graph which indicates viability, and this is the graph which indicates seed vigor. So before the viability declines, our vigor declines. So that is the most important thing that all the seed technologists should bear in mind. Just because a seed germinates, we cannot expect that everything is right, all is right. It's not so. Most of the things underlies below this, and that we express as seed vigor. If a seed germinates, it will give a seedling, but whether it is productive or not, whether it will bear the reproductive part or not, everything is decided only by seed vigor. So in this graph, what we see is the fall in seed vigor precedes the fall in seed viability. Okay, so the seed germination may be 80, but the vigor may be lesser than what you expect. That is what I want to impress upon you with this graph. So to explain more about this, we see this slide. So this is the effect of seed vigor on crop productivity. Okay, so you have the seed which is of low vigor. Okay, so you get a field like this. So that is the low plant density. Low plant density means lower yield per unit area. You will get lower yield per unit area. Then low crop growth. That means it is lower yield per plant. So individual plant will yield less, and the area as such will also give lesser yield. So this is only because of seed vigor. So if the seed vigor is low, you may get the number of plants per unit area, but the crop growth will be low, and because of plant density will be low, and you also get lower yield. So this is the important influence of seed vigor on crop productivity. So can we manage the problem of seed vigor by increasing the seed rates? Can we manage? So I expect some answer from somebody. Can we manage this problem of seed vigor by increasing the seed rates? Punita, she is my own university. I hope she answers this question. Punita, can we manage the problem of seed vigor by increasing the Hello? Okay, so this is just to break the monotony. Anybody who wishes to intervene in between can unmute yourself and then talk freely. Okay, so the impression which I want to make on you is we cannot manage this problem of seed vigor by increasing the seed rates. Okay. Hello? Jayanti?
Not increase the seed vigor by increasing the seed rate, ma'am.
You cannot increase the seed vigor by increasing the seed rate. Okay. You cannot increase the seed yield per unit area by increasing the seed rate, because
yes,
a low vigor seed will give the number of plants you require, but the individual plant will not be productive. That is what I want to impress. Thank you, Jayanti. Thank you.
Okay.
So now, what is seed enhancement? So you have the post-harvest treatments that improve the germination or seedling growth. They come under seed enhancement, and some treatments will help to facilitate the delivery of seeds and other materials required at the time of sowing. These both components will come under seed enhancement. Simply saying, the post-harvest treatments like seed coating and seed priming and seed pelleting, all this fall under seed enhancement. So they will add value to your seeds. That's all, simple. So these are the three methods which we are going to deal extensively: seed priming, seed coating, and then seed pelleting. One by one we'll see. So first, seed priming. So what is the definition or is description of priming? Priming is controlled hydration of seeds, which allows pre-germinative metabolic activity in the seeds to proceed. Okay, then followed by drying of seeds. That's all. Simply soaking and drying. Anybody will say, what's priming? It is just soaking and then drying. But it is not simply a physiological process. How? See, the controlled hydration, you can do it in n number of methods, maybe osmo priming, hydro priming, matri priming, halo priming. So I don't want to go into the details of the methods of priming. I'm going to focus only on what happens inside the seeds during priming. Basically, whatever may be the method of priming, what you do is soak the seeds, but it should be drained and dried before the radical comes out. Okay, you should not allow the radical to come out. Until then you can soak. Why? Because this soaking allows pre-germination, means botanically it is emergence of the radical. So since you prevent the soaking before this, you stop the soaking before the radical emerges. Okay, so you allow all the pre-germinative metabolic activity to proceed. The main things that happen are protein synthesis with extant mRNA, the mRNA which are already present inside the seeds. Then repair and synthesis of mitochondria and DNA. So these are the major pre-germinative metabolic activity which will proceed inside the seeds during this controlled hydration of seeds. So you are soaking the seeds not to imbibe water alone. You are imbibing the seeds to allow protein synthesis, and that includes your amylase, protease, all the enzymes. Then repair and synthesis of mitochondria and DNA. So you allow this process, pre-germinative metabolic activity, through this controlled hydration of seeds. So after that, you dry the seeds without radical protrusion. You dry the seeds. When you do this, you fix all these biochemical activities that have happened inside the seed. It will be present in all the protein synthesis will be present inside the seeds. The mitochondria repair will be existent. The DNA synthesis will be there. So you fix all the biochemical activities inside the seeds. Because of this, you get improvement in seed vigor. Okay, so this is an invigoration technique, and recently we call it as enhancement technique. Enhancement is a broad terminology. Within that, invigoration specifically deals with priming to improve the seed vigor. Okay, so this is the physiological basis of seed priming, a description of seed priming process also. So here, before the soaking, if your seed is of low vigor, after this process, it will be medium vigor. Before this controlled hydration, if it is of medium vigor, definitely after drying of seeds, it will become high vigor. So one stage improvement in priming is visible in all the seeds wherever you implement priming. Let me go to the next slide. Okay, so this is to deal in detail with the priming process. The three stages of imbibition, which allows germination, is phase 1, phase 2, phase 3. Okay, so this priming process will allow only phase 1 and phase 2. It will not allow the phase 3. Okay, so this phase 3 is normally cell elongation, division, and radical protrusion. This process we will not allow. You will allow elongation, but you will not allow division and radical protrusion. So without phase 3, if you allow phase 1 and phase 2 of imbibition, that means it is priming. You can see this graph. Yes, hello? Okay. So you see this graph, phase 1, phase 2, and phase 3 of germination. Until phase 1 and phase 2 is germination, and phase 3 is post-germination. You see this sigmoid graph. There is a steady increase in imbibition, that is phase 1. There is a stagnancy in phase 2, water imbibition phase 2. And once again in phase 3, there is rapid improvement in the water imbibition. But this phase 3 is after the radical cells elongate, that is the radical cells have come out of the seed. So in priming, what we are interested is the phase 1 and phase 2 of the germination. So during this process, as we saw in the previous slide, the solutes leak, the mitochondria gets repaired, protein synthesized using mRNAs, DNA is repaired, and synthesis also takes place. So here you see the third phase, cell divide and DNA is synthesized and radical cells elongate. That we will not allow. Okay. Okay. You see. So this is by Boolean black. So the 3 phases of priming: imbibition, activation, and growth. So this is a normal curve that we saw in the previous slide. Okay. So now priming, what happens? See, they will allow first phase and they extend the second phase. That is the activation phase is extended. We do not allow the growth phase. To avoid this growth phase, we will dehydrate the seeds. So this is dehydration or drying of seeds. Okay. So now this is the process of priming: imbibition, activation, and then drying. Okay. Now, what is the effect? So when you sow such seeds in your soil, it will just jump from the phase 1 to phase 3. It will not retrace the activation phase. So there is no activation phase here, imbibition and directly radical protrusion, because all the biochemical activities have been completed during the process of priming. And it directly goes to the radical protrusion phase. So the speed of emergence, the vigor, the viability percentage, germination percentage, everything is higher when the seeds are primed. Okay. Okay. So now these are some of the work which have been conducted in Department of Seed Science and Technology, Tamil Nadu Agricultural Universities. The work actually started from early 2003, 4 like that, and it is still continuing. We have come out with some interesting results. So we can see in this slide the effect of primed seed under suboptimal temperature. Okay, this is 20 degrees centigrade. So when I visited Netherlands, I could see that the Incotech company or other Semenis, they are seed processing all, they are all giving so much importance to seed priming, mainly because the temperature is always low over there when compared to the tropical countries. So they have a temperature stress of suboptimal temperature. Only when they prime the seeds, then they get optimal emergence of the seedlings. So compulsorily they take up priming. It is not like our Indian situations where we can do well without priming. So we feel that it is an added cost over here, but in northern hemisphere where they have the suboptimal temperature, they definitely go for priming. It is an important intervention for them. So this is suboptimal temperature. You see the control, and this is Sun-matric priming. Sun-matric priming. So I don't deal with the technology, but you can just see the effect of priming. Here also the beetroot. This is bendy. This is beetroot. You can see this is control, and this is hydropriming. You can see the length of the radicals. Then here it is mustard, control, and then hydropriming. And here it is radish. You can see the vast difference between the control and then hydropriming. So this is the initial seedling vigor. Okay, you can just imagine yourself. So initially from 4 or 5 days, you can see such vigor means what will be the effect on the rest of the crop period. Okay. So you can see this, that is temperature, this is water stress. It's very important for our own region. Our own region where we have this gambling of monsoon. So this is chilli seeds, the same tray, you have the primed seeds performing. You can see few seedlings over here. Here it is controlled. No seed has managed to emerge. But under 60% water holding capacity, when you have some more water added to the sand, you can see the control has germinated to 1 or 2 seedlings. Here, there is full complement of emergence in the primed seeds. Okay, so this is how the priming helps the seeds to withstand suboptimal temperature as well as suboptimal water or water stress. So the same thing you can see this in with brinjal also. Okay, so one experiment was conducted in JK seeds during that period, the 2005, 4-5 like that. So they took 34 quintals of hybrid rice. There was a very important necessity that they should increase the vigor of the 34 quintals of the seeds of hybrid seeds which they had. So the procedure we will now see. So they have used gelatin, which is animal-based protein, at the concentration of 600 ppm to prime the seeds. Normally, when you use only water, you call it as priming. When you use substances like this, bioactive substances, you call it as, in detail, you call it as fortification. Okay, but they are all the same thing. You allow the seeds to invigorate themselves. So this is lot segregation. So this is preparation of the solution. You can see this gelatin will dissolve only in warm water. So they dissolve it and then add to the water and use a stick to that stir it. So then now the solution is ready. The soaking starts. Okay, they soak the seeds in the bins and then they just remove the floaters. You know very well, the floaters, very low specific gravity, they have to be removed. They cannot germinate. Then later, so this is allowed for 12 hours of priming. Okay, this process 12 hours is not just a physical process. In this time, the proteins will be synthesized using the extant mRNA, mitochondria will be repaired, DNA will be repaired. So the process is happening over here. And after 12 hours, to avoid the radical from protruding, you drain the seeds. Drain the seeds, and first day you must shade dry it. And the next day, in this process you must take care that you should keep on stirring, otherwise the seeds which are in the lower layer will germinate because of the higher moisture content. So they keep on stirring. They prevent the radical protrusion because of higher moisture content by stirring the seeds, by walking over it. Second day, they put it in the sun, under the sun. So by this, we bring back the moisture to the original level. Okay, so this is priming is soaking and drying back to the original moisture content. So sun drying. So now this is the testing. So you see this is control and this is primed. So you can see the germination and vigor of control is far less than the primed seeds. So all the 34 quintals of the seeds, they were able to improve in performance by this method, and they placed it in the market, and they could follow it for 3 months until they were sold, and it was fine. There were no complaints. It was a successful implementation, practical implementation of seed priming in JK seeds during 2005 or at that range. So now we have seen practically the importance of the seed priming, the description and how the performance, but now we will deal into the molecular aspects. So here you see this graph we have already seen. The big doubt that arises is this priming process is only the activation phase. Is it only regarding the activation phase, that is protein synthesis, repair of mRNA, mitochondria, and repair of DNA, all those things which are seen. So our doubt in our Department of Seed Science and Technology, what is the impact of this drying phase, drying or dehydration phase on the invigoration process? On the invigoration process, let me take a 2 minutes gap. I invite some questions from the participants so that we can break the monotony. Anybody who is interested in giving some feedback?
Hello, madam. Good morning.
Good morning, Vikas.
Good morning, madam. You, as in previous slides, you discussed about the seed priming and its importance in increasing the vigor.
Yeah.
With respect to that JK seed where paddy seeds were used.
Yeah.
So, can you show some clarification on oil seeds, particularly sunflower, which crop I'm working on?
Okay.
So, and how long after priming, how long these can be stored and in what conditions? So that is also a very important aspect after priming. So that's what I wanted to clarify.
Okay, because it's a very good question. There are a number of articles on the effect of seed priming on seed storage. I'm happy that you raised that question. The main thing is this duration of soaking. Okay, so this duration of soaking, you should standardize it very well. When you increase this activation phase, soaking from activation phase to this growth phase, that is radical protrusion, the name of division and elongation. Okay, only then you will have problem with the storage. So you should standardize in such a way that 12 or 24 hours will be okay for most of the seeds. After that, proper drying, then it behaves like the ordinary seed, not the primed seed. Okay. In fact, this initial improvement in seed vigor will be sustained. For example, you get 10% increase in seed germination. Okay, initially. That difference in 10% will remain in your seed even after 6 months. I'm guaranteeing that.
Okay. So this is with respect, irrespective of the category of seed, means
irrespective of the category. Irrespective of the category, we have seen in vegetables as well as our
oil seeds, pulses.
Yes, irrespective of the category, it performs well. But the thing is, going before for a large scale implementation, just do it on a small scale in your laboratory and test verify it. If you get the increase immediately, you dry it and store it for 6 months, there won't be any damage. That is our experience.
Okay.
Thank you.
Thank you, madam.
Thank you. Anybody else?
Good morning.
Okay, so we'll go to this importance of dehydration phase, because this is winter school. We should focus much in detail, because what we have seen is only undergraduate level. So this is one thesis that we have took for the PhD. The treatments that we gave for this is for tomato. This is the imbibition phase. Okay, so this is the dehydration phase. And after imbibition and dehydration, this is the re-imbibition stage, how the primed seeds perform. So we took treatments such as 12 hours soaking, 24 hours soaking, 36 hours soaking, 48 hours soaking. So this is to see what is the impact of soaking duration on seed vigor. Then this is the primed seed. This is the best duration standardized for rice. So after 48 hours, you dry for 3 hours. That is one treatment. 6 hours, one treatment. 9 hours, one treatment. 12 hours. Complete dehydration. This is actually primed seed. So this is soaked and this is primed. This is in between. Okay. So we want to see what the impact of seed drying. What is the impact of seed drying on seed drying means seed dehydration phase on improvement in seed vigor. That is a priming process. Okay, for that we have seen a certain experiment. You can see. So our finding is more than imbibition. Imbibition is important. There is no doubt about it. But more than imbibition, this dehydration phase decides the efficacy of priming. How? You see this graph. Okay, so this is dehydration 3 hours, 6 hours, 9 hours, and 12 hours. So as the dehydration duration increases, you can see the electrical conductivity. Electrical conductivity is the measure of membrane integrity. Okay, so the seed membrane gets repaired. That is one of the important effects. Membrane means not the entire seed, the seed coat. This is the individual membrane, mitochondrial membrane, ribosomal membrane, nuclear membrane. Like that, individual membranes, they get reorganized. That is why finally the leachate from the seed gets reduced, which is measured in terms of electrical conductivity. So after 48 hours of soaking, if you dry it for 3 hours, the electrical conductivity is high. That is, reorganization has not yet happened. But when you dry to 6 hours, 9 hours, and finally 12 hours, the membrane integrity has been regained. This is an important effect of dehydration on the seed. Okay, then gibberellic acid. So you see, so as the seed dehydration progresses, the ability of the seed to synthesize gibberellic acid also increases. The ability of the seed, that is the vigor level increases, therefore more gibberellic acid is also synthesized. Gibberellic acid, its impact on seed germination need not be explained because this is the precursor. Gibberellic acid will directly influence the amylase synthesis. Amylase will go and break down the starch and give the required energy. So like that, gibberellic acid only triggers the seed germination. After imbibition, the first activity that happens inside the seed is gibberellic acid synthesis. Now, the level of gibberellic acid synthesis is directly influenced by the dehydration of the seeds after 48 hours of soaking. So, dehydration is an important phase. It is not done only to decrease the moisture content to the original level. It is done to complete the priming process as such. Take this condition, a 48 hours soaked seed without drying, you take up sowing. Then 48 hours soaked and then dried back for 12 hours to original moisture content, and then that also you compare. You will see the difference. Why the difference? That only we are seeing in this research. Okay, you can see this. Okay, then the alpha amylase. So dehydration 3 hours, 6 hours, 9 hours, and then 12 hours. Okay, you see 12 hours dehydration, you get 11.78 milligrams in the 3rd hour of re-imbibition. Re-imbibition means the germination process. 3rd hour itself you get, but with 3 hours of dehydration, that is more or less, it is not at all dried. Okay, for that you need 12 hours of re-imbibition. Okay, so this happens 3rd hour, this happens in the 12th hour. So you can see the fastness or the quickness of alpha amylase activity in the dehydrated seed vis-a-vis seeds which are not dehydrated. Okay, so alpha amylase activity of dehydrated seeds are higher than in the wet seeds. Okay, so this is the anatomical potential. Okay, all the biochemical activities will be manifested as the anatomical potential. So these are the embryos of the tomato seeds which were experimented. So seeing is believing. You see, this is 9 hours dehydrated seed. Okay, this is 12 hours dehydrated seed. Now, how it behaves? See, you imbibe it for 3 hours. This is the result. When it is dehydrated for 9 hours, 3 more hours of dehydration you get. So you see, these are the cotyledon, the leaves, cotyledonary leaves. The embryo, this is the cotyledon. These are the cotyledonary leaves. This is the radical portion. Okay, so the leaves have actually grown inside the seed itself. They are ready to just emerge out of the seed coat after radical protrusion. So this picture shows that the 12 hours dehydrated seed, okay, even after 3 hours you can see the leaf. Okay, but here it has not come out. It is just trying to come out. That is, the cell elongation has not happened here, whereas it has happened here. Okay, so this is the embryonic study. I mean, the studies on the image analysis of the embryo from the different treatments. Okay, so this is the protein profile. SDS-PAGE, you see, this is 9 hours and 12 hours dehydrated. You can see the band over here. This band, it is not present over here. So this is dehydration is also a shock or a stress for the seeds after imbibition. So they try to synthesize some sort of special proteins. So special proteins appear when the seeds are dried. Other things are normal. This alone, you can see special 2 proteins when they are dehydrated. So this is the research throws light on the importance of the dehydration phase, wherein the membrane organization happens, which results in lower electrical conductivity, and then gibberellic acid synthesis, then amylase activity, and protein profile. Everything culminates in higher of a primed seed. Okay, higher levigal, that is the invigoration process. You see here, this is moisture content, electrical conductivity, gibberellic acid, as well as seed viability. So we are comparing the unprimed seeds and the hydroprimed seeds. Okay, you see, this line is hydroprimed seed. So what this graph shows is imbibition itself is faster in the primed seed. Imbibition is faster when compared to the unprimed seed. So when the imbibition is faster, the water will go and do all the necessary priming activity. You see, the electrical conductivity is actually low in the case of primed seed, whereas it is high in the case of hydroprimed seed. Organization membrane is good, so that reflects in the gibberellic acid synthesis. So the gibberellic acid synthesis is high in the primed seed when compared to the unprimed seed. Finally, everything manifests in the improved seed viability and vigor. Now this is about the viability percentage. So the primed seed has higher viability when compared to the unprimed seeds. So this is to compare the hydroprimed as well as unprimed seeds in terms of moisture content, electrical conductivity, gibberellic acid, and seed viability. So now this is our concluding slide for this importance of dehydration phase. So 48 hours imbibition alone has been completed. Here 48 hours imbibition and 12 hours dehydration has been accomplished. So in germination, here there is 3% increase, here there is a 19% increase. Vigour index 26% increase, 93% increase. Gibberellic acid 278% increase, 355% increase. Amylase 80% increase, 225% increase. Protease 190% increase and 87% increase after dehydration. So this is the importance of drying. In most of the research papers, you would have come across a term called as on-farm priming. On-farm priming is nothing but you stop with this imbibition phase alone. You don't proceed to the dehydration phase. That is, the imbibed seeds will be directly sown in the land. Of course, it has some effect, but when you dry it and sow in the field, then you get the highest manifestation of priming effects. Okay, so this is about all about priming and the research that we have. Okay, so what do you take home from the series of slides that you have seen? Repair and invigoration process initiates during imbibition phase, but it culminates, it completes and culminates only during the dehydration phase. So priming is not just imbibition. Imbibition followed by dehydration is priming. So the basics are over. A small break. You can take some questions.
Good morning, madam.
Good morning.
Have you validated the seed priming, hours of imbibition and hours of dehydration in pulses, particularly green gram and black gram?
Sir, green gram and black gram, they are very tricky seeds. They are proteinaceous seed. You cannot soak for 12 hours, sir. We have done priming. 3 hours is sufficient, sir.
3 hours of soaking.
Soaking, yes.
And 12 hours of dehydration.
Yes, sir. Normally 12 hours drying is sufficient for all the seeds because within 12 hours they will come back to the original moisture content. If need be, next day we can go for sun drying, not on the first day itself.
Thank you, ma'am. Very nice presentation.
Thank you, sir. Thank you, sir. Any other question?
Morning, madam.
Morning, sir.
Yeah, this is regarding the okra priming. You told it's only hydro priming?
Yes, sir. Hydro priming only.
And also maybe
12 hours soaking, sir. 12 hours you soak it and then you can dry it back to original moisture content, especially okra. If you go to the ISTA rules, they specify that the seedling evaluation can be done only on 21 days, sir. 21 days. But in our conditions, it can be done after 1 week itself. But if you keep it in the germination room where you maintain the germination temperature around 25 degrees centigrade, okra will not germinate fast. But under optimal condition, it will germinate fast. Okay, sir. This okra has a special behavior. Tropical conditions is more suitable for okra when compared to the temperate conditions. So instead of keeping in the germination room, if you keep outside means germination will be far better, seedling growth will be far better when compared to the germination room, sir.
Yeah, okay, maybe. My next question is most of the priming are just a pre-sowing treatment, and maybe how is the next follow-up as far as the storage is concerned?
Sir, this question I answered to another participants also, no problem, sir. This is a regular doubt among all the scientists. Seed storage, whether it will be affected by priming or not. The question is relevant, but the duration of the soaking, if you extend into the radical protrusion states, that is the phase 3, then definitely there will be an impact, sir. So you must be very careful in standardizing the duration. I said that for the pulses, 3 hours is sufficient. If you prolong to 6 hours, then you will have this poor storage. But in the case of tomato brinjal chilies, you have to soak it for 48 hours, sir. 48 hours. So it depends on the species. Proteinaceous seeds, they absorb the water very fast. The germination process is also fast. So if you do the priming process properly, standardize it, then definitely the improvement that you see in the initial stages itself will be stay put in the seeds for another 6 months, as we have seen in most of the crops that we have studied, sir.
Yeah, okay, thank you. It's very clear. It's the timing of the priming is very, very important, and also
Yes,
the
Priming
dehydration.
process, yes, sir. Yes.
Dehydration, maybe.
Yes, sure.
Thank you.
Thank you, sir. Thank you, sir.
Madam, one more question.
Okay, sir.
Madam, in case of vegetable crops and other things, we used to recommend from the microbiologists that go for a seed
Pseudomonas.
treatment of Azotobacter and KSB, PSB likewise.
Yes, sir.
And then if we soak the seeds and after priming, if we give the treatment, then germination will be faster?
They have their own effects, sir, because these microorganisms, they get embedded inside the seeds and they have their own effect. There will be additional effect, definitely, sir.
Because what they recommend is before sowing 1 hour earlier, give seed treatment. So what I'm asking is, seed pelleting is different or seed treatment is entirely different?
So seed pelleting is entirely different, sir. You forget it, sir. You can compare seed coating with the priming. What you say is just before sowing, you want to make a slurry treatment with the pseudomonas.
Sprinkler syrup nahi.
Yeah, that in that instance, there won't be priming effect. There won't be priming effect, but these organisms will have their own effect. They will enter into the seeds or stay superficially on the seed, and the colonies can multiply and they can have their own effects, sir. Definitely. But if you add the organisms in the soaking process itself, that will also be good.
Okay.
Okay,
Thank you.
sir. Thank you, sir.
Madam, good morning. Very nice presentation.
Not able to hear what sir is saying.
Hello, ma'am.
Yes, sir, I'm not able to hear. Now it is okay, sir.
Madam, I have one question.
Okay.
Is there any difference between seed hardening and seed priming?
What is the difference between seed hardening and priming?
Yeah.
Okay, sir. Sir, actually, the seed hardening is not the terminology to be used. Russian scientist Henkel actually introduced the term pre-sowing hardening, sir. There no seed comes. But the process of soaking and drying was introduced by some person called Will, W-I-L-L, Will H. During 1883 or so, and it was followed by Henkel, Russian scientist, during 1940s. He made an elaborate paper about this seed pre-sowing hardening, not seed hardening, pre-sowing hardening.
Yes,
but that is the basis for this priming. During 1970s, early 1970s, Heidegger brought out this term osmopriming. That was a thing where they soaked the seeds in polyethylene glycol, mannitol, like that.
Yes.
There they wanted to extend this activation phase so that this synthesis process can take place. But later on, what people brought out in many research was, we need not go for this osmopriming. We can go for soaking in water itself, but control the hydration of, radical protrusion. Originally, Heidegger, osmopriming, what he put forth is, if you soak in PEG, that is polyethylene glycol, the radical protrusion will not happen because of the osmotic relations. The imbibition will be very slow, so radical will not come out. That is controlled hydration that he proposed. But later on, even in hydropriming, physically we can control this radical protrusion by stopping this priming process, sir.
Okay.
So water itself is sufficient, but you can also take up some bioactive chemicals, as I explained in the case of gelatin. It will have its own effect, some 2-3% improvement. There is a term called seed fortification, where you use this panchagavya, this gelatin, or any substance which gibberellic acid, whatever it is, which are bioactive and they can improve. So that is fortification. This is hardening. Now everything is coming under one terminology: seed priming, sir.
Priming.
Okay.
Okay.
Hello, ma'am. Hello.
Hello, sir.
Thank you, ma'am. Thank you.
Thank you, sir.
Hello. Hello.
Hello. Yes, sir.
Yes, this is Dr. Vishwanath from Onion Garlic Research Center, Pune, ma'am.
Good morning,
I have one doubt. Use of this like bio agents like Panchagavya, like even like Jivamrut.
Yes,
What
sir.
is the mode of action, ma'am? Like if we recommend for seed treatment or some soaking. What is the real mode of action and how to treat the seeds? Can you give me insight, ma'am?
Sir, this Panchagavya or Jivamridam, whatever it is, sir, a sort of fermentation is allowed, sir. Fermentation is allowed, and that will lead to synthesis of enzymes and hormones, whatever it is that we should actually see only further research, through further research. But the thing is, more bioactive substances, maybe enzymes or hormones. So they will be synthesized. You imagine if you soak the seeds in gibberellic acid, you get an effect, sir.
Yeah.
So similarly, the microbes will make some fermentation process and release most of these bioactive substances. When you soak in the seeds inside that, then you get that fortification effects.
Ma'am, one more doubt, ma'am. For this onion, what duration do you recommend for priming, ma'am?
Onion. So we will see in the coming slides, sir. We have actually standardized for most of the crops.
Okay, ma'am. Okay. So thank you, ma'am. Thank you.
Thank you, sir. So now we will see some of the slides, research activities which we have conducted.
Hello.
Hello, sir.
Madam, I have one question.
Hi, sir.
Madam, one of my MSc students is working on rice, and he has used aromatic and short grain varieties.
Okay, sir.
But madam, what happened due to some rainfall? He is not getting that entire grain after hulling and milling. Means that head rice record is very low. So, madam, can priming be beneficial in this case?
So you say that the priming, no, sir, seed filling is itself a problem here.
No, not seedling. After harvesting, before harvesting there was some rain, madam. Late showers. So we are getting head rice record very less.
Okay.
So can it be beneficial? Priming can be beneficial for it.
So you could not dry the seeds. No, sir, that is the problem. You could not dry the seeds properly after harvesting. That is why you are getting low vigor, isn't it, sir?
It might be, madam.
Might be.
Yeah.
So priming, normally this internal activities will be boosted, sir. But in this case, where drying is not proper, some pathogen effect could be there, sir. One thing. Then overheating could have happened, and that could have damaged the seeds, sir. So artificially, you are making the seed stressed because of higher temperature and higher microbial growth. We can try, sir. There may be improvement, but we have not done such work. You can try in a small scale, 1 kg, 2 kgs. You can just take the seeds and subject to priming. But the main problem in priming, no, sir, I must accept, soaking large volumes of seed and then drying back. That is the biggest problem, sir. That is why priming is not adopted in large scale in our country. Wherever need arises, you can go for priming, but by default, like coating, you cannot do priming because the logistics involved is too much, sir. But for high value seeds like vegetable seeds, it is possible, not for our low volume, low value seeds like cereals. Yes, sir. Please continue.
Madam, moreover, what happened last year? Our farmers, due to late shower, there was a huge loss because some grains were generated on panicle itself. So, madam, is there any experiment conducted for that the germination should be prolonged in the field itself?
Sir, actually, sir, we have tried to do some research for this in our own laboratory by spraying this sodium chloride, which is a dehydrant, sir, actually. But I don't know how far it will be practical under continuous rain. But if there is some anticipated rain, then we can go for the dehydrant. Another paraquat, I think, sir, one herbicide which kills the herbs by dehydration, sir. So when you, I don't remember the research result now, but we have done it in our department. When you spray that herbicide on our crop during this maturation, that is after physiological maturity, to enhance the dehydration of the seed in the plant itself, you can go for that spray so that the seeds will be dry enough to harvest and then minimize the requirement of drying. This pre-harvest sprouting can be avoided, sir. But priming cannot be a solution for this problem. Okay, sir.
Thank you, madam.
Thank you, sir. Okay. So a new priming technology was developed in our department by this student, Chandan. He belongs to Karnataka. Now he is working in National Seeds Corporation. So as I said earlier, the soaking, draining and drying is the biggest problem encountered in priming. So he took up the research on using a washing machine for this seed priming process, and he developed a process called as spin priming technology. So here it involves soaking of seeds, automatic draining of the seeds, and then spin priming for surface drying of the seeds. So you can take out the moisture by the spinning process, and then you can dry it easily later on. So he found that there was an improvement over the conventional priming by giving higher increase in seed germination as well as seed vigour. So he has analyzed for different crops like tomato, brinjal, chilli, bindi, onion, beetroot, carrot, cauliflower, radish, etc., etc., etc. So you can see the difference. I will just skip on very fast. It is just to show that how this spin priming is better than your conventional priming and then the control. It is in this order. You can see this is 4 minutes spinning and this is control. This is 5 minutes spinning. This is ridge cord. You can see the root and the shoot growth. Goats respond fantastically to this priming, especially spin priming. So this is bitter gourd. You can see the root and the shoot. And this is carrot. Very good difference. So this is anatomy. So this is the control. You can see this tetrazoleum has been used to stain the embryo. The area of staining and the intensity of the staining is much higher when compared to the control in the conventional priming as well as this spin priming. You can see the leaves have actually grown so much. It's only because of elongation, probably not by division of cells. So this is the bendy, you can see. So it means that the mitochondria has been repaired and synthesized and more respiration takes place. That is why there is intensity of the staining. You know, dehydrogenase is the enzyme behind the staining. When the mitochondria is more active, then your dehydrogenase enzyme activity will be higher. When the dehydrogenase enzyme activity is higher, intensified. So radish, you can see the leaves are actually expanding like this. Once you open the seed coat, you can get like this. So this is onions that you can see, sir. Somebody asked onions 24 hours hydropriming followed by spinning for 1 minute if possible. Even without spinning 24 hours, if you can soak it and then dry it 1 day in shade and next day in sun, that will be fine. Onion responds well. Okay, so we have standardized for all these crops. So advantages of the spin priming: automatic draining of the seeds, surface drying through spinning, plus better improvement in seed germination and vigor. So I think we completed the discussion earlier. If you have any doubts on this, using this washing machine for spin priming, we can just have a few questions.
Hello.
Hello.
Hello, ma'am.
Yes, ma'am.
Yeah, I am Dr. Chitra from IMBIBITION, New Delhi.
Good morning, madam.
Good morning. Good presentation. Ma'am,
you.
what is the cost of this spinning drum or whatever you have shown?
This is just washing machine, regular washing machine, which we use in our house. The top loading washing machine. It might come around 15,000 rupees today.
Okay. That is spinning speed, everything like it will vary with different machines.
What we used is around 300 RPM, like I think, but you can stabilize it yourself. So we used 1, 2, 3, 4, 5 minutes for all the crops, and we standardized the duration of that spinning also based on the machine. Completely, it might vary. Sure. So what machine you use?
Yeah, definitely it will vary. But you have mentioned the spinning time. That RPM also, if you can give means.
I remember it is 300 RPM.
300 is low only, no? Okay.
Yeah.
Okay, you can confirm for like for uniformly it is for all the crops or it varies.
No, that RPM we cannot adjust in the washing machine.
That is there.
Standard,
but no RPM with different like 500 or 500, 700,
yes,
800.
But
RPMs
I understand.
Are there.
I understand. I agree. This washing machine which we used, that is only 300 RPM.
Okay. Good one.
Hello.
Yes, sir.
Hello, madam. This is Rakesh Matat from US Raichur, madam.
Good morning, sir.
Good morning, sir. Good, madam. I want to know whether is there any lab model or tabletop model of priming unit is available, madam, anywhere?
Sir, actually, the priming unit as such, there is one model that I have seen from this seed processing Holland, sir. That is a very big machine, and they will also price in a higher side only. But what locally, indigenously, what we suggest is you can use your washing machine, top loading washing machine for this priming process. You can soak it. You can tie the seeds. If you take 1 kg seeds, sir, you make it into 500 kgs. I mean 500 grams and 500 grams. Two bundles, sir. 2 bundles. Then you soak it in your bucket with water or even the washing machine itself. Then
there is an option called spin and rinse or something like that, no? In washing machine.
Yes, that's all, sir. You put it inside.
I got it.
Yes, sir. You put it inside and then give the spin timing. You have your watch. Sir, you have your watch, sir. For 5 minutes, you allow the spinning. 4 minutes you allow, 3 minutes you allow, 2 minutes you allow, and you put it for germination. You will see the difference, and accordingly you can standardize it, sir.
Okay. And another thing is we have to put in some net bag, no? Net bag it is better because
cloth bag itself is sufficient, sir. No problem.
Okay. Which type of bag is there, madam?
You can use cloth bag, sir.
Okay.
But since it is a centrifugal type, you have to put into 1 kg means not just 1 kg, sir. You put it into half kg, 2 cloth bags.
Yeah, usually the washing machine is at least minimum is 5 kilo. This one is there.
Yes, sir. Yes.
5 kilo. It starts from 5 kilo, I think.
Yes, sir. 5 to 8 kgs we can use, sir. That is best.
Okay, ma'am. Thank you, ma'am.
Thank you, sir. Okay, sir. Hello, sir. Hello, sir.
Madam, nice presentation.
Thank you, sir.
Hello. Madam, I have a question. I am Prashant Panda from Odisha.
Okay, sir. Good morning, sir.
Good morning. Madam, what is the priming time?
Priming time, sir?
Germination time varies according to crop.
So this is the priming time, sir. You can see the slide.
What is the time of soaking? Means how many time we can soak a seed?
Yeah, so tomato means 48 hours, and you can see the slide. Tomato, brinjal, chilli, 48 hours soaking, sir. Carrot, 36, onion, 24.
I want it in pulse crops. Means pigeon pea, mung bean, urad bean, these crops.
Sir, for all the pulse crop, it will be around 3 hours only, sir.
3 hours.
Yeah, because you know it will be faster imbibition, no, sir.
It will germinate. Otherwise, if we keep in many hours, then seed may germinate.
Yes, sir. So
Yeah.
not more than 3 hours, sir, for pulses. That is sufficient, sir.
Acha. With 4 minutes spinning.
No, sir. That we have. That I don't have the data now. We have done it, but most of the people don't require. You can just mail me, sir. I will give you the.
In mung bean, urad bean, we don't need priming, but in pigeon pea we need priming.
Sir, any crop, if you prime means there will be a substantial difference there. The only thing whether we have the time and then labor and all those things. That's the thing. But any crop responds to priming, sir. There is no.
All crop response, but a hard coated seed responds more.
All crops responds more only, sir. But whether it is required or not, we have to decide, sir.
Okay, madam.
Okay, sir.
Thank you very much.
Thank you, sir. Thank you.
Good morning, ma'am.
Good morning.
This is Devanusi Datta from Assam Agriculture University, Assam, ma'am.
Okay, madam.
Very good presentation, ma'am.
Thank you. Thanks.
So, ma'am, I have one query. During your spin priming technology in the washing machine, is there any chance of seed damage because you are using 300 RPM?
Yeah.
So is there any chance of seed damage, ma'am?
Yeah, see, madam, you can see the slide. We have standardized it for goats and all, no? 3 minutes, 4 minutes, but for chilli only 1 minute. So likewise, you have to standardize it. If you increase the time of spinning and you change the RPM, maybe there is some damage. Otherwise,
okay, ma'am.
The limited amount of spinning is actually good for your seed. When compared to the conventional priming, you get an improvement over that. Why? Because madam, spinning is also a stress, you know. This stress, no. I think it influences some protein synthesis or like that heat shock protein madri. Some proteins will be synthesized. Therefore, spinning to that limited extent is better than your conventional priming. It is only advantageous, madam.
Okay, so ma'am, this timing is standardized by your university, ma'am. Spinning
yes.
time.
Yeah.
Okay, ma'am. Okay, thank you so much, ma'am.
Thank you. Thank you. Okay, shall we go on to the next technology? Okay. So this is the seed coating. So we have completed priming successfully. Now we are passing on to the next technology, that is seed coating. You can see the picture over here. So this coloring, originally it was called as coloring. Now we are calling it as coating. So the seed coating, what is a seed coating polymer? You have so many brands in the market. Incotec is a market leader, and Bayer Seed Growth is an international brand. Littles is a local Tamil Nadu based polymer manufacturer. And Hitron, our own Professor Dr. C. Dharmalingam, after retirement, he started a company called Hitron. He got a patent, and then he is successfully in the market by selling the seed coating polymer. Okay, so what is the seed coating polymer? It's a product in which additives, I repeat, additives are dissolved or dispersed in a liquid dyed polymer. Okay, dyed in the sense it is colored polymer, and it is used for coating or spraying on the seeds, followed by drying. So here also you require drying. So this is the polymer. So polymer is an adhesive. Okay, so it will be a colored adhesive with necessary additives. That is actually seed coating polymer. I'll show you a video. It is about coating of seeds. So this is the polymer dispensing unit. So the seeds are rotating and the polymer is sprayed over them and it is getting colored. So there is a polymer mix. It is sucked and then sent inside. Bendy seeds. So this is so you can see the difference between this is the bendy seeds, normal bendy seeds, and this is pure coated seeds. So this is coating. That is a drum which rotates at a given RPM, and then the polymer will be sprayed on it so that it gets coated like this. So what is the purpose of seed coating? The purpose of seed, it is an adhesive. It helps to coat the seeds with the fungicide and then the insecticide, and plus it also improves the color of the seeds. When the seeds are during harvest, as somebody was telling, it rains and that there is some discoloration of the seed coat, but the quality is not affected. In such case, you can use this polymer seed coating to improve the appearance of the seeds. So originally, to include the fungicide and insecticide coating, we used to have some slurry treatment using water. So there used to be dusting off and some pollution problem also for the person who treats it. But when you use this polymer coating, such problems can be avoided. It is very efficient, and the color of the seeds also improves. Then there is uniform distribution of this fungicide and the insecticide all over the seed. So now I wish to introduce to you the TNAU seed coating formulation. This was the research taken up by my own student, Mr. Karthi, in the year 2017. So normally the advantages of seed coating polymer is it enhances the appearance, it acts as a carrier for nutrient additives, it's used as an adhesive for insecticide, fungicide, it ensures uniform application of active ingredients, it prevents dusting off of active ingredients, but ours is an extra and most important effect. It improves seed germination and seedling vigor. None of the products in the market are as effective as us. They claim, but ours is giving better improvement over seed germination, seedling vigor. Those things, why it is giving also, we'll see in the slideshow. So this is the control. So this is formulation 1, 4 grams per kg of seed. This is formulation 2, 4 grams per kg of seed. You can see the root growth, compare it with the control. So this is maize. So this one is so good. And this is sorghum. And this is soya bean. You can see the difference. So likewise, we have standardized for 21 crops: rice, maize, sorghum, pearl millet, finger millet, black gram, green gram, soya bean, groundnut, sesame, castor, cotton, tomato, brinjal, bendi, chilli, onion, bittergourd, maygourd, ripgourd, cauliflower. So this has been done via 2 theses. First year, one student, Sandhya, worked on it, and then the formulation was finalized by the second student, and then we standardized for all these crops. So this is the dosage, and this is the amount of water that you should add 8 grams for tomato, 8 grams of the polymer along with 200 ml of water, like that. So what is the effect of the formulation on the shelf life of seed? We have seen, you can see this is after 6 months of storage. This is cotton. So this is controlled and this is your coated seed. You can see this is the root growth is not normal because of some other factor. So it has not germinated, the control has not germinated, but here, better God, it has shown some germination, good germination in fact. And this for snake god. Drought also, there is some effect. You can see in this tray, this is number of seedlings of the coated seeds, and this is control. You can see with control, compared with control, the speed of germination and germination increases under drought conditions. So this is for sorghum, the same effect you can see. So we have done it for black gram. So black gram has turned red gram because of our polymer coating. So you just have to see the plant morphology. So you see this treated seeds, and this is control. See the root growth. So the number of the seminal roots, secondary roots are so rich. Root hairs are so rich when compared to your control. So because of this, you get a very thick stem, and the leaves are also thick and green, and the dry matter obviously has to be higher here when compared with the control. So we took up 2000 kgs of maize seeds from a private seed company to improve the coat and then improve the seed germination percentage. So this is the measuring of the formulation. This is the process. We did it along by help of our own students because they got exposed to this coating and seen everything by this. So this is the covers which were given to us, and all students are working. We accomplished it, and the coating process. So this is the difference. This is the control and then the coated, germination, root shoot, etc. There is around 10% increase in germination. So just because of coating, this is not priming, this is just coating. There is 10% improvement in germination, and there is corresponding improvement in root shoot as well as dry matter production. The story does not end there. So this is 10% improvement in germination maize. And you can see the thing is here, not just the germination increases. You can see the vigor. The area covered by the leaves is higher in the coated seeds when compared to the ground coverage. This is the control. Okay. See, the control germination is 81, but when you count the good seedlings and weak seedlings, 67% good seedlings, 22% weak seedlings. This is the weak seedling. So like this we had 22%, like this we had 67%. So in the field, the number of productive plants was 57%, 59% in the case of control, whereas in coated seeds it became 83%. So it increased the vigor of the seeds, so that the number of the weak seedlings produced was reduced to 7, whereas in the control it is 22. So here 93% good seedlings and 83% were productive. You see? So this is the difference in the seedling vigor of the same seed. This is controlled and this is coated. You have to see the plant height, stem girth, leaf area, root volume, root hairs, and then root sheath. What does root sheath mean? When you pull a plant from the soil, the volume of soil that adheres with the root is called as root sheath. So here when you pull, it comes with the big volume of sand when compared to the control. So you can see the height and root. Okay, so this is how the coated and the control. You have the number of the roots, but here you can see that it is so dense when compared to this. The volume area, the area occupied by the control here and the coated here. So this were transmitted to the farmers also. So they used their own technology. They don't have the machine, therefore they used their own technology, and they could see the improvement in the seed vigor. So one farmer, he came to the department and then got it primed and coated onion for onion. So he is the farmer. So exclusive benefits of our technology: higher seed germination, improved seedling vigor, better storage life, and improved stress resistance. So this technology, this is my student Sandhya, 2017, Karthi, 2018. So the MOU was signed on 15/2/18 with a private company called Vriksha Agro Ventures, and they have taken up the commercial production and supply of this particular polymer. It is called as Shidacot. For the farmers, the diluted version N.Vidai, they are marketing this in Tamil Nadu, and this is all over India. So now, any questions? So if there are no questions, we'll pass on to the next.
Hello, one question.
Hello, ma'am.
Yes.
Maybe as you mentioned, the 10% increase in the germination, how do you justify the increase in germination by seed coating? It's only the effect of polymer or something else? Because
yes, sir. Sir, actually, polymer has its own effects, sir. Mainly, it influences the imbibition of water. That is the first factor, sir. So we took n number of polymers from the industry, and we standardized which one is good. By that, we standardized the best polymer. Later on, we added some additives also. One of them is gibberellic acid. So polymer has its own effect, and the additional effect is because of the gibberellic acid, sir.
yeah, it's like some sort of invigoration along with the coating.
Yes, sir. Yes, sir.
Yeah, thank you. It's good.
Thank you, sir. Thank you, sir.
Hello, ma'am. I'm Chitra again.
Hello.
I'm Chitra.
Yes, Chitra.
What is the seed storage life after this coating in the groundnut? You have mentioned about groundnut and paddy, isn't it?
I don't think we have done groundnut, but of course, there is improvement in groundnut also, no doubt about it. But our studies mostly, we have not done groundnut, I think. But another one occasion we have done for groundnut, and it has performed well. Always people say that it is a very thin testa, therefore it might get damaged.
Yeah, that's why I'm asking.
But what the farmers did is they will put inside a gunny bag and then shake it so that there is no much damage to the seed coat. Okay. In fact, if there is a research also, I have the photographs also, unless and otherwise you damage the radical portion. Radical. Yeah. Seed coat does not have any additional effect to the germination. In fact, if you remove the seed coat without damaging the embryonic axis, the germination will be better. In the groundnut seeds which are dormant, the dormancy is only because of the seed coat. There are studies also like that. Therefore, per se, seed coat is not important for germination. But embryonic axis should not be damaged in this process.
Damaged.
Okay.
Okay.
Hello, madam.
Yes, sir.
This is Dr. Vijay Kumar from US Raichur, madam.
Okay, sir. Good morning, sir.
Madam, my thing that polymer, their ingredient, what are the formulations which is being disclosed, or it is not disclosed, madam?
No, sir, it is not disclosed, sir, because we have signed that MOU with that private company.
Okay.
They are commercializing it, sir.
Okay. But in that NSP workshop, we have discussed that none of the companies are giving disclosure of this content of the polymer.
They will not give, no, sir. That is a trade secret. They have invested lots of money in R&D. Therefore, they will not disclose, sir.
That's a okay.
Okay, sir. Thank you, sir. Okay, shall we pass on to seed pelleting? Okay, so this is the third subject that we should discuss. Seed priming is fully physiological. Seed coating is thought to be physical, but TNEU seed coating formulation has some physiological effect also. But pelleting is full and full, it is only physical. It does not have any physiological influence over the seed. It is the difference. What's the difference between the seed coating and seed pelleting? So seed coating, it will not change the shape, size of the seed. And weight increase will be only up to 10%. But pelleting, it will alter the size, the shape, and the weight of the seeds. It will increase the weight by 8 times or even 10 times, depending on the size of the pellet. If you put 8 kg, then that will become 80 kg. So logistics is the biggest problem in pelleting. Also, the pelleting time is very, very lengthier. In the case of coating, it can be an online process. But in the case of pelleting, it will take at least half an hour for each batch. If it is pan coating, it will take 3 hours to complete one batch. So it is not at all possible to integrate in this seed industry. I mean that processing plant. Now, only this, what to say, European countries, they are very fond of pelleting because there they have a different soil type. They have a different climatic conditions. They are fully automated. They cannot use the seeds, raw seeds directly on your seed drill. Therefore, they go for pelleting. In our condition, there are not much takers. In the early 2000s, many of the seed companies took up pelleting. When the Incotech company just set their foot on the soil in Ahmedabad. But now there are not too many takers about pelleting. But let us see the difference between the coating and pelleting and how it can be used. So benefit, it aids in mechanical separation of the seeds. This pelleting will help in the mechanized sowing. Then small and irregular shaped seeds can be handled easily. Then seed wastage can be prevented. So these are the benefits of seed pelleting. Precautions, there is a danger of getting multiple seeds inside a pellet. Then empty pellets also, there might be chance. Technologically, if you are not properly implementing it, these problems will come. Surface may not be smooth. Then faulty technology will lead to breaking and dusting. So standardization is very important. Size, internationally accepted size of the pellet is 3 mm. If you take it 5 mm, it's not going to be feasible. Less than 3 mm, it's not going to be useful. So it might also have an influence on speed of germination as well as seed germination. Pelleting, you imagine on 1 gram of seed, you are adding a weight of 8 grams of seed. Therefore, it's a burden for the seed. Therefore, it might affect the speed of germination and seed germination only if your technology is faulty. But many companies have standardized a good technology. So speed of germination and seed germination is not mostly affected, 90%, 99% it is not affected. In TNAU also we have standardized the technology and we have been very successful. So this research I have been taking up from 1996 onwards. That is when I entered into this career. So at that time, this was my small experiment. I used a carpenter to make a pelleting frame and the pellet puncture. And this was the first pellet that I saw with my own eyes. At that time, the Incotech has not set their foot in Indian soil. So based on what we have studied in our theory, so I tried to make this pellets for sesamum because we face lots of problems with the sesamum spacing in the field. Therefore, we tried it. But later on, we changed all these things with the help of JK Seeds. We got a project and we changed this size, shape, and all those things. So this was our original pelleting expedition. So spraying using this small biochemistry sprayer for use it for your what to say chromatographic studies. So you put it in a box and then you get pellets like this. It's nothing but spraying, dusting, spraying, dusting, spraying, dusting alternatively. So you can see a video now.
बेस आहे तो आफ्टर 50 years
So this is Dr. C. Vanitha. She was working as an SRF then. Now she is in NSP also. She is an assistant professor in Department of Seed Science and Technology. So all this work was done by both of us. It was a very interesting period. So we actually conceptualized this machine. It was our own brainchild. Got it made through a mechanic in our own place near Coimbatore. And everything was done during 2008. Later on, it was followed by a student called Celebration. Originally for JK seeds, we worked only for mustard, sesame, tomato, brinjal, chili, carrot, onion, radish, amaranthus. Everything was standardized by Celebration and Menaga 2017. You can see the pellets along with the seeds. So the germination also very good. For brinjal, especially compared to control, there is improved germination and growth. That was an astonishing fact. Onion and carrot. So there is no delay in speed of germination. It's a very small seed carrot, yet there is no delay in speed of germination. Germination percentages also as good as the control. Amaranthus and tomato. So this is amaranthus. Seedling growth, it's also fine. So this pelleting mixture actually, Incotech Netherlands, they were in contact with us. Their powder, they sold it in India, 300 rupees per kg at that time, 2008. Ours came only up to 30 rupees per kg. It was very, very cost effective. So they wanted us to send 200 kg to test in their facilities across the globe, and we sent it. But it did not materialize. But later on, during 2017, it was licensed to MS Reliable Corporation Chennai, but they are not able to successfully market it because there are not too many takers in India for seed pelleting. But benefits of the seed technology developed by our department is good speed of germination, no reduction in emergence. It's a domestic technology or indigenous technology. It is cost effective, yet it meets the international standards. So this is our lecture. I must thank you very much for patiently being with me through this entire presentation, priming, coating, and then pelleting. Yeah, I have to only few more slides. So based on the technologies that we developed, we conducted proficiency training on seed vigor and quality enhancement techniques for seed companies, a 3-day paid program. So many seed companies participated: Dharani Seeds, Highwedge, Haryana, Maha Seeds, Egoriaman Seeds, Riksha Agro Ventures, Sungene, like that. Many companies took part, and they were given hands-on training about all these techniques which were developed in our own laboratory. Yeah, this is the lecture, and this is the field exposure. So I wish to acknowledge Dr. C. Vanitha, Vageshwaran, Malarkodi, Menaga, Sandhya, Karthi, Chandan, Silamarasan. They are students. They are all the people who worked along with me for this proficiency training, as well as other research works also. So finally over. Thanks for your patience. We have crossed 115 slides. Okay, we can take questions now.
Thank you so much for such a nice, very informative, and also we had in between very lengthy deliberations, ma'am.
Thank you, sir.
Permission of speaker. If any questions are there from participants, means you can ask and unmute yourself and you can ask questions to the Umarani ma'am.
This is Devanusi again, ma'am, from Assam.
Yes.
In case of seed pelleting, you are using 2 powder mixer, ma'am.
Yeah.
What are those, ma'am?
So once again, it is a technology which has been commercialized through an MOU. And
okay. Okay.
Thank you.
Okay, thank you, ma'am. Thank you.
Good morning, madam. Sorry, good afternoon, ma'am. Hello, good afternoon, ma'am.
Good afternoon, madam.
I am from Odisha, OUT Bhubaneswar, and I am working as seed production officer in case of vegetables.
Okay.
Madam, in case of okra, so, madam, we have 3 years old okra seeds, and I had also gone through hydro priming, but I could not get any result. Any other priming technology or method?
No. What is the actual germination percentage of your lot, madam?
Okay.
Originally, what is the germination percentage?
No, madam, these are the aged seeds.
Aged, right? Okay. Whether it germinates up to 40%, 60% like that, you should have some idea.
Nothing, madam.
Nothing means it is all dead. See, madam, priming will work on low vigor seeds, not on dead seeds. Dead means it is dead only.
Okay, madam. Thank you, madam.
Okay. Any other questions?
Okay, if there are no questions from participants, means now I invite Dr. Nagendra Kola sir to propose vote of thanks.
Thank you, sir. Thank you, Dr. Dhananjay. Dear participants, Dr. Umarani Madam, Professor of Seed Science and Technology, Tamil Nadu Agriculture University, Coimbatore, has delivered very good talk covering detailed information about opportunities and perspectives of seed quality enhancement techniques in vegetables and flower crops. It is very informative, valuable, and with very nice presentation. Madam, you are such a wonderful presentation along with showing practical demonstrations through videos. Through these presentations, our participants got more benefited practically. So on behalf of all our participants, organizers, College of Horticulture, Bidar, University of Horticultural Sciences, Bagalkot, I extend a wholeheartedly thank to you, madam.
Thank you, sir. One information I want to share, sir. Recently, I got a patent for the secondary protein foliar spray. And Dr. K. Parameshwari was the senior research fellow working along with me, and she is one of the participants also now. So in your presence, I wish to thank Parameshwari for her support and then being along with me during that big process. So I saw her not only in the presentation. So I wish to register that she has put a lot of efforts to complete that work, sir. So I also thank each one of you for your patient hearing. I hope it was of some use to all of you. Thank you very much, sir.
Thank you, madam.
Thank you.
Take over to Dr. Dhananjay.
Thank you, sir. Thank you, Nagendra Kola, sir, for proposing vote of thanks. Madam, thank you so much for accepting our invitation and delivering such a nice lecture, ma'am.
Thank you, Dhananjaya. Thank you for providing me this opportunity. Thank you.
Thank you, ma'am. Thank you so much.
Thank you. Welcome.
And also, I extend my profuse thanks to all the participants for your patient listening to
patient hearing. Yes.
Yes. To this lengthy session. In the afternoon, we have a session at 3 o'clock. I request all the participants to rejoin by using the same link. Thank you, one and all.
Thank you. Hey, Ami, sulle. Thank you.